Thank you for joining the workshop. I'm the chair, Masanori Arita from DDBJ, Japan. And first, I have to apologize for you that the information about this workshop was not listed properly in the, almost until yesterday. But we will have 5 speakers from major data repositories in the world. And the first speaker will be Mano from Metabolomics Workbench, followed by, followed by Claire from EBI, Metabolites, and then we proceed to Massive, KBDS from Korea, and DDBJ. So this is workshop, not research presentation. So I really encourage you to question and ask. And my original intention is to establish a federated repositories over the world, covering the all continents. So if you are coming from countries like Latin America or Africa, I really encourage you to join this kind of a network so that we can share all data from across the world. So let's start. Please, Mano.
It's fine with me. We can email the slides or any format is fine.
Is there a shared repository for this meeting, or how can we share our slides? Does anybody have an idea for sharing our slides? It's okay for us, for example, the DBJ, to share all materials with you. But
because everyone's registered, who's at the workshop, so we could probably get all of them. Might be possible.
Yeah, I think the key thing is all speakers are happy to share. We'll just find out from the organizers how best to do that the most efficiently. So we'll take that as a takeaway and then let you know.
Great, thank you so much.
Thank you, Professor Arita, for introducing us. Thank you. So I'm Mano Moria. I'm from University of California, San Diego, which maintains what is known as National Metabolomics Data Repository, also as a Metabolomics Workbench. I will be talking about some aspects of data sharing, analysis, and integration with other types of data at NMDR. So it's important that I mention that pretty much all of this work I will be presenting is actually done by the core team of Metabolomics Workbench that is headed by Professor Shankar Subramaniam, who couldn't come here, and actually much of the work is actually carried out by Dr. Owen Fahey, whom many of you might already know from previous meetings and so on. And this work is really a culmination of effort over 20 years, which started with lipid maps and then continued into Metabolomics Workbench. So at the cellular level, metabolites are essentially more like endpoint readouts for functional functions in physiology and pathophysiology. For example, going through a series of processes such as transcription, translation, enzyme activation, and so on. Eventually, that reflects into changes in the levels of metabolites, which are also easy to measure in serum in many cases, and they are often directly affected by environmental influences such as nutrition or malnutrition based on exercise and then whatever medications we take. So some of the challenges in metabolomics are, how can they be measured accurately, how quantitative those measurements are. How can one analyze the omics measurements data and interpret results from metabolomics experiments? How can we organize, store, use, and share metabolomics data? How can this data be integrated with other types of data such as genomics data, transcriptomics data, and proteomics data to get an overall picture of the system's view of the biological system? And most importantly, what are the implications of metabolomics for systems medicine? Can we metabotype an individual? To address some of these questions, several years ago, NIH started what is known as Common Fund Metabolomics Program, which resulted into National Metabolomics Data Repository, for which Metabolomics Workbench is the portal shown on the left-hand side here. The right-hand side, the table on the right-hand side shows a snapshot of the different aspects of the metadata that has been deposited at NMDR. For example, how many total studies are deposited there, how many of which are public. For example, as of now, we have around more than 2,800 studies which are open for public. Then we have almost more than 280,000 samples or biosamples related to these studies, and a number of other statistics which gets updated almost on a daily basis. So overall strategy for metabolomics data repository is shown here. For example, starting with experimental data, when a metadata submitter deposits the data using an online system, if the data includes named metabolite data or targeted data, then it goes into a database based on a number of metadata information that the metadata submitter specifies. And once the data is into a relational database, we have a suite of tools which can be used to analyze data from various studies. In the case of untargeted data, the metadata is put into the relational database, whereas the actual large-scale data such as raw master spec files and so on, they are processed through another pipeline, and eventually we have a corresponding set of tools which can help do some of the analysis of that data as well. So over the last 12 years, we have seen tremendous growth in the number of studies that have been deposited, and that pace has not slowed down, and in fact, we expect it to be at least maintained or grow even further. So one of the key aspects of saving, facilitating sharing this metadata is the use of what is known as MW tab format or MW format, which is actually sort of based on UniProt format, where we have three main fields. One, the first field corresponding to metadata, where one can specify the project and study-related description, and other information such as what is the subject, information about the treatment that was carried out and how the samples were prepared and so on. The second part is the actual data itself, whether it's a named metabolite data or in the case of untargeted data, we retain some useful information, for example, in which file it has been saved, etc. And then we have additional information. Besides MW tab format, which is human readable, we also provide this information in JSON format, which might be easier to exchange and reuse in various programming languages. In fact, both there are MW tab format parsers available in Python programming, and also we have a validator for those who want to submit their metadata in MW tab format itself to begin with. So the Metabolomics Workbench website or the portal for NMDR, the main website looks as shown here. We have the ability to browse and search through all the studies. For example, we can get a summary of all the studies and then find more information about the studies. For example, we can search on the basis of what are the species or organisms for which that study is related to, what is the disease for which this study is related to, as shown on this slide here. For example, we have seen that majority of the studies are from humans and then mouse followed by rat. And in terms of disease, we have the highest representation from cancer. So we can find a lot more information about a particular study, such as exemplified here for the very first study that we had submitted. We have a number of tools that we can apply related to some basic analysis, such as the composition of the measured metabolites corresponding to different metabolite categories. And then we can do some basic statistical tests such as t-test and look at it as a volcano plot, and going to more advanced analysis as a correlation analysis. and others such as the partial analysis, squares analysis, etc. Besides MW tab format, another very important aspect of metabolomics workbench data is the use of what is known as RefMet nomenclature for metabolite names. It's a unifying nomenclature. It tries to use the most common name for metabolites. For example, on the next slide, we'll see that there is quite a bit of variations used when researchers submit metabolomics data. This reference provides us ability to perform comparative analysis across different studies. And it also helps with integrating it with information on biochemical pathways. Currently, there are more than 170,000 unique metabolite names in the RefMet database. Just to give an idea, when researchers submit metabolite names, there can be a humongous variety, and it's really important to harmonize those metabolite names so that any subsequent analysis of the study or its integration and reuse with other tools is feasible. One of the important statistical tools at Metabolomics Workbench is called Metastat, which has kept on growing over the years based on the feedback from users and metadata submitters, where we have the ability to search metabolomics data, both data and metadata, based on the metabolite name, the species name, disease context, etc. In the recent times, we have had a lot of emphasis on the ability to share the raw data itself, have that feature available at Metabolomics Workbench. So in terms of total raw data, we have almost 50 terabytes of data in NMDR. Submitters are encouraged to submit the raw data, for example, in MZML or MZXML format, etc. And now we have the ability to actually browse through the individual mass spec data, and a number of tools provided by other researchers. For example, just to name one tool from Professor Peter Dostin's group, we have the ability to use that, facilitate connecting to that tool from the Metabolomics Workbench itself. And going forward, this is going to be even more important. For example, if new tools become available to validate mass spectra or identify new approaches to identify unknown metabolites, then being able to share this raw data is really going to be critical. So I think I already covered some aspects of this. For example, here for all the data, raw data from individual files submitted by the researchers can be actually visualized and using appropriate tools. Another very important service in terms of how we allow researchers to fetch data from Metabolomics Workbench is the use of REST interface, where we allow one to find metadata based on metabolite names, study ID, disease context, etc. So I have shown here some examples of the format we use for the MW REST interface. But this is not complete. There is a lot more, and based on the feedback, we are further expanding our MW REST interface to make sure that as much as possible, much of the metabolomics data can be fetched by researchers. Considering the emphasis on being able to integrate metabolomics data with other data, over the past several years, NIH has started another program called Common Fund Data Ecosystem, which as of now is essentially a collaboration, very tight collaboration between about 12 to 15 large-scale repositories corresponding to different types of data, and this is going to further grow. And starting this year, now they have come up with 5 integrated centers, for example, Data Resource Center, which we are actually going to take care of, and then cloud related to cloud computing effort, and then Center for Training, and so on, just to name a few. So this is the So we every 3 months we deposit our metadata into the common fund data ecosystem portal, so that we provide a way where people can search data not only on metabolomics but many other types of data as well, so that down the line they will have a better ability to figure out in what ways they can integrate that data. So with that, I would like to end my presentation and would be happy to take any questions. Thank you.
Okay, we can take questions for each presentation, but do you have any from the audience?
Yeah, that last slide was very interesting. I wonder if you could elaborate on what other resources are contributing to that through the NIH.
Oh, you mean which other groups are contributing?
Yeah, to that. Which other resources in the NIH are given metadata to be able to search across?
Yeah. So we have one of the largest scale programs called GTEx, where they have data on transcriptomics from different tissue. So they also submit their metadata. So we have the ability to search that metadata as well. Then we have data related to extracellular RNA, and then genomic variation type of data, just to name a few. But let me go back to that slide. If you go to the portal, that is info.cfd.cloud, there across the different tabs, for example, in this screenshot, you can see the second tab from the left that provides information on different common fund programs, where we have detailed information about what kind of metadata they are providing. And going forward, we are also allowing these centers to deposit their tools, analysis tools, in the forms of APIs. And we are encouraging formats like open API format, as well as REST formats, so that people will be able to analyze their data. And in some cases, we are also we have processed data of different types, so that because often in order to analyze and integrate raw data in raw format, it can be quite challenging for the end researcher. But if we have processed data, then first of all, the center that processed it can account for the variation across different studies on which they have the data. So from that viewpoint, going forward, we have more representation of processed data as well, and we plan to make a number of tools available. So this effort actually actively started this year, and it will continue at least for the next 4 or 5 years, and hopefully quite a bit beyond that.
Okay, please use your mic sound.
So thank you for the presentation, very nice. And I would like to ask a question about the metastats. Just quickly had a look through the web interface. So if I understand correctly, would it also provide the human metabolism pathway that the metabolite species will participate?
So if I understand the question correctly, you want to specify a metabolite name and find out whether it can tell which metabolomic pathway it belongs to.
But there's a problem is some of the, let's say for example, the amino acid metabolism, for example, the aromatic amino acid like tyrosine, which I've just noticed on the web interface, could also be metabolized by microbiome as well, right? So would you consider like adding some more options about the microbiome metabolism pathway as well?
Sure, yeah, we are very open to the feedback, and based on the feedback, in fact, in the last few months itself, we have added more aspects to both the metastat as well as the MW REST interface. So please do write to us. The email address is at the Metabolomics Workbench website, and we would be happy to see what we can do.
Yeah,
Thank you very much.
Thank you very much.
Okay, let's move into the second talk by Claire Donovan from EBI Metabolomics.
Thank you.
Okay. So I hope you don't mind, but I asked for the lights to be turned on because I quite like to see people when I'm talking to them. That's a personal preference. And that's the microphone. So, this one.
Okay.
Yep. Thank you. Okay, so I'm very glad to be here to talk about this, and I really encourage you to engage. We've run this workshop a couple of years now at the Metabolomics Society meeting, but we really need the community to help us understand what you want from us and what you need from us so that we can develop it. So I'm from, well, lovely picture, that's the building, lovely gardens. If you ever come to visit us. But what you might not know is that we're part of a much larger organization across Europe. So this talk is really going to be about how we can be global. So I put up this slide to show you that we're part of a large ecosystem within Europe. 29 member states. We're not the European Union, but we're kind of parallel on that with a lot of scientists doing various things. And the one that I work at is the European Bioinformatics Institute, which is based in Cambridge, UK. So no labs. We're on a site with labs, but we are the bioinformatics people. And we do various things. But key things is deliver data resources for Europe, and we train people from all different backgrounds. And that's what I really, really care about. So this is the original mandate for the setting up of the EBI. So EMBL is 50 years old this year. The EBI is 30 years old. And believe it or not, I was there at the beginning. And this was the original email sent out to say, this is what we plan to do, which is that the data is conveniently accessible. So for all of you who are too young, weren't even around back then, people used to fax us sequences. It was a moment when we moved into browsers and submissions. So I've been through all of this process. So I feel your pain when you come to submit things, but honestly, you have no idea how better it is now than it used to be. But our complete evolution over those 30 years is to continue to deliver to everybody. But the other thing I would really like to stress, so even when we put out that release from the mothership in Heidelberg in Germany back then, DDBJ was already our friend here in Japan. So it has been an international collaboration for a very long time, and it was GenBank in Washington. And I've been very lucky to work quite a lot in Washington and also with DDBJ in Mishima. So I'm very pleased to be here in Japan for this meeting. So this is a slide from our official slide deck, which is to say that we've always wanted to do global. So we do serve our community, but the only way, and we've had this right from the very beginning, is that we have to work with people all around the world. And I'm personally passionate about it. And it's one of the joys for me working at the EBI. We have 800 people, roughly, and it's 40 nationalities. So it's a super cool place to work. You might be misled by my accent, but I'm not British, I'm Irish, and in this little part of Cambridge, it's really nice to be international. We're surrounded by fields. So the key thing, as I say there, is that we have a shared responsibility with collaborators in the US, Japan, and Europe, and we'd really like to get to a place where we do that for metabolomics as well. And that's why I'm really passionate about this particular workshop. There's also key things that, and this is for those of you who come from the global south, we really need to push on and make sure that the data is used by everyone for the benefit of everyone. And you might not know, but the EBI is on a campus, which is funded by the Wellcome Trust, which is a big, huge charity. And that is their mission, and they're already asking us to train people all around the world, and I'm very happy about that. We also do work always finding new funding models, because sustainability and resources is a key thing. We flagged up their Global Biodata Coalition, and again, this is trying to ensure that it isn't just the big countries that have fancy resources, it's resources all around the world, and that we all help each other to develop for the benefit of everybody. So I really hope, I don't know how many of you are experimentalists. Okay, that's great. I'm really glad you're in the audience, because I don't want to talk to the people who already know you should be data sharing. So how many of you do actually data share on a regular basis? Not as many as I would like, but I'm also really positive that the rest of you are in the room to learn about it. And hopefully, by the end of this workshop, we can convince you it's worth your while, even if there's a pain point at the beginning in learning about it. But the key thing is it does actually benefit you. And it will increasingly, as you see from my colleague previous slide, the NIH is insisting the data is shared. Other funders in Europe and around the world are insisting, but key thing is that the journals do. So if you really want to get your tenure track position, pushing up your citations, you need to share your data. Just as a top tip. So for how many of you have heard of FAIR? That's better, but still work to do. This will haunt you in the future if you don't find out about it now. But you can become the guru in your lab and know everything about this, and that will also raise your profile. So please investigate. We will share the slides after, but feel free to take as many photos as you want, as long as they're not of me. Thanks. So this is our resource, but I'm not really going to go into the details of it. I'll put up a slide at the end where you can find us. We have 2 poster sessions later this week, and both curators and software engineers from the team are here, and they'll be available to talk to you about any questions you have about metabolites. But for the purpose of this talk, I really want to talk about the metadata and how we can all work together. Why I really care about this is because while we're the European resource, a recommended repository, our submitters are actually global. And as you can see, our growth, like all the Metabolomics repositories, is growing exponentially. And also the type of data is changing. So initially, when it was set up in 2012, it was very plant-based. But now it's more than half of it is human. So again, the database has to continue to evolve, and as my colleague said, they have a lot of cancer data. We have a lot of other kinds of data, which reflects the people who submit to us. So we don't have any mandate. You can submit whatever you like, but that is a challenge for the repositories. And we need to have the experimentalists to help us continue to develop the resource so we capture it. So we're not just thinking about plants. We have to think about everything. So that's the key thing about metabolites. But all my years of experience in bioinformatics supporting life science resources is that there's niche areas, there are particular databases, and if any of you are interested in setting up a database, I'm not telling you don't. Just make sure you learn about it, that you reach out to those who are already doing it, and you fully understand what you're taking on, and we'd be happy to help. For instance, next week I have a call with a new Metabolomics resource in India, and we're happy to share all our code, we're happy to share all our metadata. That's the key thing. Data sharing is not just you giving me your data. We're happy to give stuff back if that's what you need. And that can be just in your core facilitator at your institute, or it can be if you're going out on your own. One of the strengths that I think we had in developing metabolites was ontologies were already very strongly in mind. My background before I took over metabolites was UniProt and the gene ontology, so I've been doing this a very long time. So that was how we were able to ensure that metabolites could scale. and that our metadata is rich. So this is I'm just putting in a metabolite slide here for the point of pointing to it. But if you see there the descriptors, protocols, samples, all of that, when you're submitting to us, we suggest to you the ontologies that you should be using so that we can narrow it down to exactly what you think you want to tell us, and then your data is comparable with other people. So all the submitters are saying the same suggestions. Obviously, if they're not there, there is an opportunity to let us know in that submission series, and then we submit those terms to the relevant ontologies for you and standardize afterwards. So it's an ongoing effort. Ontologies never finish. That keeps lots of people in work, so not to worry. The other key thing is about the protocols. Those of you who attended the reuse workshop yesterday that we were also luckily part of, is that there's, for true reproducibility, you really do have to have all the details. And I don't know if Michael Wittig is in the audience or not, but he's been really helpful, and his team in Germany, with helping us ensure we work really hard reading vendors and companies' manuals to make sure we're capturing exactly the right chromatograph tube and everything like that. The curators really focus on it. We'd like to automate that a lot better. But Michael has been really helpful in telling us exactly how much of that is reproducible. And that's an ongoing piece of work. The other area where I think, and again, that will only improve as long as more and more people get in touch and submit diverse data to us. And it also helps you when you come to learn about a study or how to do a study, that you see how someone else has actually done the experiment. And in the MQuack meeting just before this one, we learned about how they did an analysis of the papers, and it was really hard for people to know exactly how they did the experiment. They had the concept of the experiment, but not the actual detail. And that's really sad. You've read the paper and you don't know how to do the experiment. So in a way, the databases can be repositories for those protocols. So you don't have room in the paper. You really want to talk about all the exciting science you did. You don't really want to talk about your column. Then the repository can be the place to put that. But under the other key thing is the compounds, identifying what the metabolites are. We also learned about targeted, untargeted, how a lot of the features in the raw data are not actually identified. Having the data available in the repositories enables ongoing identification. And that information goes into the compound libraries. And this is another area that our community really needs to work together to make sure that these libraries are available to all, not just the countries that are rich enough to pay for them, or the labs that are rich enough to pay for them, and to benefit everybody. So when it comes to sharing, you've already heard from my Metabolomics Workbench colleague. You're going to hear from my Metabobank colleague shortly. We do have two ways where we currently exchange data. So it's called Metabolomics Exchange, but it really is just a portal. We automatically push things. This was set up a long time ago, and it hasn't been developed very much recently. We'd really like to develop it. Omics DI is another one that was funded by the NIH. And that's where all the different omics are put together. But again, there isn't very few common standards. And so it's really just an index. It is an easy way to find out if your paper is there, if the paper you're interested in is there, and then if the data sets are all available. But again, we can do better. And this was a forum where we would like you to tell us how to do it better. So to throw a little challenge you a bit. We firmly believe in learning from our colleagues who have done it before us. Metabolomics is one of the newest omics. So Proteomics Exchange has been running for a while. And as you can see, it is international. And we many of the... So Pride is at the EBI, JPOST is here in Japan, Massive is our colleagues in the States, and there's other partners. But what's really nice is this has been running for a while. They've learned the lessons, and so it's really great to talk to these people about how we can do better and not replicate the mistakes they've already learned from. Always a key. I don't believe in reinventing the wheel. 30 years in bioinformatics teaches you that kind of thing. So how it works, it was originally set up in 2013. They have a common data identifier and a common data model. They're mostly not replicated, so unlike the genomics data that until very recently used to be copied every night from Japan to America and America to England, they keep them in separate place. There's also a consortium agreement, so if one resource runs out of funding, the others agree to take the data. This is really, really important because if you're not aware of this, resources, even ones at very important or very established institutes, money runs out. And it's a key thing that your data doesn't disappear. So this has worked in Pride where, for instance, I'm not going to name it, but a database disappeared in Canada and others picked up the slack. And this was, I really care that for the future of metabolomics, we get this clear from the beginning so that your data doesn't disappear. It's really, and then there's common submission guidelines and common portal for public sets. And then this is where I really like, and it's quite similar to what you might know if you've ever submitted genomics data. Each institute, each database can have their own bells and whistles, their own flavor, their own specific specifications, but the common things to make sure that they still talk to each other. So in the case of Proteome Exchange, it's the experimental mass spectrometry and then the metadata. And you can do other things as you like. Those are optional, depending on the priorities of your country or your institute. And that's how I hope it will evolve in the future. Oh, sorry. So, final up with, these are the people who found us right now. Thank you very much. This is the team. I didn't get organized enough this morning to pop their names on there, but if you look up our website, they're all there. And then, as you can see, I'm right there at the front, but I'm retiring come August. Lovely to meet you, but I'm off. But I'm still passionately enthusiastic about all of this, which is why I'm here when I could be packing up my house. And I also want you to, the first two people, our second person there is Callum Martin, whom some of you might have seen speak this morning. And the fourth one along is Thomas Payne. He's also here. He's the head curator. And then at the end is Juan Antonio Vescancio, who's the current head of Pride, and who will be taking over Metabolites as soon as I leave the building. And he has graciously agreed to come here to meet all of you so that we can ease the transition. I may have bullied him a little, but he's here. Okay. So just to finish up, like I said, if you want to talk to any of us, we'll be at the poster sessions later this week if you want to talk about metabolites or anything else. Okay, that's me done for now. Any questions?
So thank you very much for an interesting talk. I'm just curious, it's a two-part question. I'm just curious, do you track the use case of the data?
Yeah, so that's a really good point. So also at the EBI is a thing called Europe PMC, which is a cousin of PubMed in the States and probably other international collaborations. And so what we're working on with them, I did it manually, going through papers that quoted metabolites accession numbers, and they were the ones that were just the depository ones. And then there was papers that actually took a metabolite study, did a reanalysis, and published another paper, or they used it to drive their research. And what we are doing right now is that they're going to automate that, so that it won't be us looking for that in the future, and it will be exposed on our website. So it was a beautiful way to spend a few days looking at how people are reusing the data, but it's important to automate it.
Yeah, so I also tried to do the exercise because I was curious, but maybe this is also a call for the community. My observation is that the use case for metabolites or repositories are somehow focused on the bioinformatics side of the things, like how, developing new methods for metabolite annotation and identification, but not really on developing new biological insights.
Yes.
And I believe one of the reasons for that is that people putting data in repositories don't put the actual disease metadata, right? So I don't know if this is from a child or this is from a man, a woman, what other comorbidities. So we see in papers that when they do cancer, no cancer, they adjust for certain covariates and confounders, but the data for that is not there. What would be nice is if we can, let's say, answer a question about diabetes from a data on cancer that was uploaded, but that's not possible for the moment because most of the metadata I've seen in Metabolomics Workbench or metabolites are really focused more on the method and how you can annotate the metabolite, but not the disease.
Yeah, so that's a very valid point. So to address the first part, when I did the review of 100 papers, more than half were for benchmarking or development of new tools, but there were ones that were more about the biology, and I'm happy to share that list with you if you like, because it was painful to do it. I'm happy that it's not wasted. To address the second point, the curators in our team, and I'm sure in all the other resources, do encourage people to give us as much metadata regarding to the disease and the biological context, but a lot of people are still scared about what it is that they're safe to share. And so I know Metabolomics Workbench has a controlled access facility, but I'm not sure how much that people kind of apply for that. But the EBI has just started with proteomics and metabolomics. We're going to do a federated model similar to what the EBI already does for genomics in the UK. So originally the idea was that there would be just one repository at the EBI, but that didn't fit with a lot of our countries who have nervousness about purpose data, and that's absolutely appropriate. So what they've done now is they've made it federated so that it's mobile, so that it can go to Finland or it can go to Spain, but that the metadata standards are the same and can be exposed on the portal, so you can query to see which data sets you want to access. And then it's up to you to reach out and get the data. That we can't help with. Did I help? Okay, great. Oliver.
Hello, good morning. So I am following this trend over many years. What happens in the repositories and integration of data sets becomes more and more important. We have heard just from the Metabolomics Workbench that they used a RefMet tool for internal conversion of metabolites into identifiers. They invented their own identifier RefMet ID. But other repositories do not use the same RefMet ID. So the question is, how can we better integrate data across repositories, not only for, of course, identified compounds, but also for unknowns. But first, for identified compounds. Even in RefMet, not everything is completely referenced to each other. And if you then look at other sets that are deposited elsewhere or that are published elsewhere, you will find that there is a huge discrepancy in terms of ability to even say that these three studies looked at this overlap of compounds. So how does the EBI handle that and collaborate with, say, PubChem?
Okay, so there was a lot of questions in there, so I'm going to address them one by one, and I'm sorry. if it takes a little bit of time. So yes, that is a critical thing. I didn't put a slide in because I thought I had less time than I thought. But our ontology that we use is CHEBI, which is chemicals of biological interest, which is originally developed very much for proteins. So they came at it from a different perspective. They were thinking more about ligands, as ligands in my protein is binding this. We submit to them, and they enable us to cross-reference to lots of other databases, including PubChem. So we have an official collaboration with PubChem. PubChem pulls in our data. We push it out to them. I would encourage you to explore CHEBI because it is mapped to lots of other resources, some of them automatically, but some of them manually, including RefMet. But then we come back to my whole point, and I really want to stress that we have to have an ecosystem of resources. Dr. Owen Fahey, very nice guy, Irish Mafia, I can say that because he's Irish too. But he's also super, super good at lipids. And our curators at the EBI, not so good at lipids. So they kind of focus on all the other ones. So what would be really great, and we are hoping to set that up with Lipid Maps collaborators in the UK, in Wales, that they do a brokering service so that when things are submitted to us at the EBI in Metabolomics study, we can farm it out to our lipids experts in Wales, which is a little country next door. And they can then push it back to us. So our metabolite studies are improved, and their database of lipids is improved, and obviously then RefMet is improved because it's all an ecosystem. There are other efforts that we'd really like to get involved in. We just haven't done it yet, like the Universal Spectral Identifier, which then is more hardcore. But I'd like actually, because you're responsible for such a resource as well, Oliver, where do you think we should be putting that effort? Pardon?
Well, I'm not in charge. I don't have the funding resources to tell you what to do or not to do. But obviously, there is a demand for many, many, many years to do these identifier exchanges. Mm-hmm. And there's the only exchange that works to some extent is the PubChem exchange. And that is having its own problems. So the people are a little bit, waiting.
Yeah, well, stop waiting and send us an email saying this is what I want and this is what I need, and then not me any longer, but my replacement and my colleagues in the different resources, we can write the proposals both to our institutions and to our funding bodies to say this is what the community needs. And this is something that I really, really want to stress to everybody in the room. If you don't ask, you don't get. Okay? And we're not mind readers. We come to these conferences, we try really hard to find out what it is you want from us. But if you write us an email, especially if you're in a rant, that's okay. Can't do this because I don't have this. Just send us an email. And then we'll do, and I'm sure our colleagues, I see nods from colleagues in other resources. If you don't tell us, then we can't help you. Hi.
Thank you, Claire, for your wonderful talk and great service to the community. My question is, what happens to the sample, the data when we submit it to vendors, when we receive only a intensity table and they have the raw data, but we actually use federal money to buy those services? Is there any way to encourage them to share the data?
Well, I can share, and I can definitely say this because I'm leaving. I can tell you that when I first took over in 2017, I couldn't believe this situation because I had come from genomics, proteomics, where data had been shared for a long time. I was around when people were trying to clone genes, but they were slapped down on that really quickly, so we never had to really deal with that after that. I did haunt people like Biocartes and Metabolon, and in the end, they did collaborate with us. So we do have a pipeline with Metabolon, which they all charge you for, and we get no money, just saying. That's not the bitterness be showing. But the only reason that changed was not my cheerfulness and me calling them or talking to them. It was because the NIH refused to fund the grants until Metabolon agreed to share the data. It is only the MZL, not the full raw data, but I believe they're changing that policy shortly. So I would encourage whatever federal government you have going on, tell them the story. I'm happy to follow it up with an email. And that's what works. It's the only thing that works.
So,
yeah, that would be ideal too.
They will change quickly and provide the raw data.
And the other thing is to work with, there's a lot of institutions in all of our countries that have core facilities, including my home one in Heidelberg. So we do have metabolomics labs, they're just not at the EBI. And reach out to them. You don't, there's an ecosystem. But I'm fully supportive of industry trying to make money. It's great, they employ people, that's all great. We just need to find a way that we all benefit.
so the NIH data, open data policy, sharing policy, is we share those data. If we have only an intensity table, would that be helpful to load?
It doesn't fit our requirements right now, but I would still encourage you to, there are other repositories like Zenodo or wherever, not to waste your data. It's still worth something, and it promotes that discussion within the community, but I would also encourage you to follow up with your federal, or if you can find another vendor, I don't know how practical that is, but do talk to your government representation. And if the EBI can help in any way with that, just let us know.
Thank you so much.
You're welcome.
Well, one addition from me to the question by Oliver about metabolite nomenclature, we need to collaborate with journal publishers. It used to be that publishers had their own nomenclature system. For example, journal-wise, they had a guideline for describing metabolite names. For example, our journal does not use alpha-ketoglutarate, but we use oxoglutarate instead, something like that. But nowadays, there are so many journals, and especially open access journals, do not put any regulation about metabolite names, so that everybody can use its own his or her metabolite names. But we database system need to collaborate and actually have as publishers so that they follow our guidelines. That's really critical in my opinion.
Yeah, there's another thing that when, and again, it's something that the resources could talk about, is that internally with, I mentioned KEBI, what was a really nice part of that resource is that the curators have manually annotated a lot of the synonyms. So even when a submitter gives us a name which is not the official name, we have an automatic pipeline that pushes it through and matches the official KEBI one. That is also code we're happy to help share, and other resources we can match up on that.
Okay, let's move to the third talk. Thank you, Claire. Now we come to Yashin El Abiyad from Massive GNPS.
Okay, hello. My name is Yassin El Abiyad, and I'm working in the lab of Professor Peter Dorestein, and also partially now with Ming-Sung Wang at UC Riverside. And I'm working a lot with the GMPS ecosystem. So this is also one of the repositories. You have already heard about quite some of them today, so here are some. you have learned that you can upload data to Metabolites, Metabolomics Workbench, also to GMPS or Massive. You can upload your raw data. And the principle sounds very straightforward, right? Like you upload your data somewhere and then it can be reused. So yeah, let's go and reuse them and the end, and now you know what you have to do. It's of course not so easy in the end, right? Because there's not really a lot of knowledge of established workflows out there on how would you even reuse this data, how would you even approach this. So this is what I would like to focus on with this talk here, to give you some kind of starting point of how you can even access this data efficiently, how you can find them, and yeah, where we want to move with this kind of things. So yeah, let's talk about the how. How can we reuse this data, and first, how can we access them, and how can we access them easily? So this is one of the projects that we have been working on, which is this central hub from which you are able to access all the repositories. So for now, it's GMPS, Metabolites, and Metabolomics Workbench data, and you have them all accessible at this one central access point. So this is one of the goals. So this is where you can just go and find them all. Of course, what do we mean by access? So the first thing that you might want to do generally is if you see one of these identifiers, so you see some, can you see my cursor here? No, you can't, but I've put it in red. So here you would see some of these identifiers that people would use if they publish one of their data sets. So for GMPS, you would see these massive IDs. For Metabolites, MTBLS. For Metabolomics Workbench, you would see these ST identifiers, and you will find them in papers sometimes, right? And by that, you can tell that somebody made the data publicly accessible. And at the GMPS Explorer, you will be able to access this data very straightforwardly at this one access point. So if you would go to this web page, you would see something like this. And you can see here on the top side of the screen that we actually put here a Metabolites identifier for a data set. And you can already see the description of this data set here. You can also use here, for example, for Metabolomics Workbench data set. Now you have a different description. If you would scroll down a little bit on this page, you would at some point get to something like this, which are something that we call USIs for these data sets. And if you look at them a little bit more closely, they look kind of like this. They always have kind of the same structure. So you have here this data set identifier, and you have here the file path to the actual file. And you can now use this within the GMPS ecosystem to have access to this specific file, also to other files within this ecosystem. So one tool that you can, for example, use is the GMPS dashboard. So that's something you would use if you wanted to have a brief look at some of the raw data that are in the public domain. So somebody has put this identifier into their paper. You now get to the USI, and now you just want to check out quickly some of this raw data, right? This would be one possible thing you would want to do. So you would just copy this USI from the Explorer that we have just seen into this, which is what we call the GMPS dashboard. You can see up here, I've already copy-pasted that in. And you will then be able to quickly look at the raw data without having to download anything, without having to navigate all these different APIs or things like that. You can extract chromatograms, you can look at spectra. And one of the things I also want to highlight about that is there's also this nice button on the top of the screen, which is copy URL to this visualization. So if you extract certain chromatograms, for example, or if you visualize certain spectra, you can click on this link. This will give you a URL, which you can use to copy, for example, into your papers. And then people can see this visualization for this specific raw data file even later when they even later after you published it, right? So if you wanted to share a certain view and a certain chromatogram, people can really see this within your raw data, which I think is much more powerful than if you just copy some PNG into a PDF, which is sometimes really hard to contextualize in a broader context. So yeah, you can put it into papers, reports, and things like that. Then one other way how you can use these USIs is that you can use them for other GMPS tools. So one thing that we have is a molecular networking that you might have heard about before. So you can directly reanalyze data on the platform. I won't go into the details on what exactly that is, but the only thing I want to point out is that you can use all different kinds of tools that we have available, and they're quite some at this point in the GMPS ecosystem. And without any downloading, you can just reuse them from any of these repositories that we have made accessible at this point. Secondly, it's nice to be able to access data, but of course we want to also have them kind of useful, right? And one big thing about that is always metadata. It's much more powerful for these public data sets if you actually know what they are and if you can search for them in an efficient way. There are, of course, some problems with that, which I would like to quickly go into. So first, the place where we make this accessible is actually the Redo dashboard. So you can see here the URL, where we have basically a huge resource, which is a harmonized table of all the metadata across all these repositories. Because one big problem for having different repositories, it was already mentioned a few times by the people who talked here now before me, is that when you look at the different repositories, it's not very harmonized regarding metadata. If you have blood plasma, it's sometimes called blood plasma, sometimes plasma. It's written in different ways. It's given to you on different metadata fields. So it's really hard to find data in these public resources. So yeah, one of the pains we went through there is to actually harmonize all these things. So we have one metadata table where we have everything in the same field, and things are always written in the same way. So you will find blood plasma always as blood plasma. Also, the terms that we use were not just like random terms we came up with, but we used already established ontologies. So here you can, for example, see some terms from the Uberon ontology. So this is a hierarchical system where you have all the different terms here, for example, for body parts, and you are able, and we are then able, or you are able, if you download this data, to put all the data into this context. So if somebody writes brain and somebody else writes something about forebrain, then these two things will be connected over this network. And if you were to reuse this kind of data for some machine learning or any other data science things, you would be able to connect this more easily. We also do this for, let's say, more exotic categories. So here, for example, if you're interested in environmental samples for sediments, and for all kinds of other fields. And yeah, after we did that, we were also able to do some kind of statistics across these different repositories of things we are now actually seeing now that we have this harmonized metadata. So here you can see all the files that we were able to harmonize metadata for the different repositories, and we can now see how these repositories developed over the years. So for example, you can see for all raw files, that for all of the repositories, the amount of raw data was increasing over the years, obviously. And what you can also see very nicely is that the different repositories have actually different kind of communities that they serve in the end, right? Because everybody has kind of different interests. And as a result, you will then have different kinds of samples that are deposited. So for example, you can see a lot of human blood plasma samples in Metabolites and Workbench. There are a lot of plant samples in metabolites, apparently, and we have a lot of different NCBI IDs, which are like different organisms in the GMPS repository. So there are quite some differences. Here's one more example on that that shows how well these different repositories actually complement each other. So here you see for mouse and human, which are our most common sample types that we have, for the different body parts, that we are able to cover much, much more if we actually put all these repositories together. And as I mentioned before, you can just download this table if you're interested in this. This is like all the metadata that we have now available for these repositories. It's at this Redo platform. That's just a small button, download database, and then you can just download a TSV and go from there. There's also an interface here, but this is a little bit outdated. I would recommend just download it and work from there. We will do some updates on that in the future, but this is not done yet. You can try it, but it will be a pain. Just be warned. And with that, I'm already like going towards the end. So just as a quick summary, first, like we already published a preprint on this. So if you want some more details, you see up here where you can find it. We are able to access now all these different raw data that we have in the repositories at the central access points. We can use it in different tools, like that. I put the dashboard link, for example, here, which you can also use in your papers and reports and so on. the metadata we have also harmonized, and you can access them and reuse them and find data this way. And yeah, as all the other presenters that have been standing here, I of course also want to say, please share your raw data. This is highly valuable to everybody in the community. Like, I didn't really talk too much about what you can now do with this raw data, but there are actually way more things you can do with that. Here are some example tools, some of which we have published over the years. So microbe mass to identify microbial metabolites, plant mass to identify plant metabolites, quite some other things. I won't go into details here. If you're interested in more, here like some self-plugs for within our groups. We will present some of these things in our talks and posters that we will have. Of course, also my PI, Peter Dorstein, who is here in the room, also will have a plenary today actually about this. So if you're interested in how you can actually use this data, he will talk about a lot of the opportunities that you have there. And yeah, with that, I want to thank you for your attention, and I'm very open for questions. So keep them coming.
Very nice presentation and great work. So you've done a lot on metadata harmonization. I wonder, have you any ideas how we can get the community to help with this? Like, is there any ideas about community curation, retrospectively?
Like how we can get the community to like, I think the first thing that would be great is to get the community to even like share the raw data. Like, repositories went different ways there. Like, some require metadata to be shared, some don't. In GMPS, we went the route of not requiring that, but of course the community can help. I mean, many, I think, upload also to this one for the simple reason that they don't want to put the effort to do it, right? But yeah, the community would of course help a lot by still going through the pains of doing that. So that's the first thing. And then secondly, just use the harmonized terms. We already heard before, there are often terms recommended that you should use for certain things, right? Like, you should call blood plasma in a certain way. So always using these recommended terms. Yeah, I would just summarize it, really, like, just go through the pains, like, read what the repositories want from you and follow it once. The second time will be already much easier. Yeah, that's how I would summarize it.
Thank you. Yeah, great work, as always, if I may. I think it's the wrong approach. To redo things is wrong. We have to get the correct metadata when data are being acquired. So that is not the fault of GNPS, Metabolites, Metabolomics Workbench or so. It's the fault of the people who acquire the data. Because that's the only time when you have the attention of the client who wants metabolomic data for their study. Then they will answer what exactly there is to answer. So the question is, and these are the metabolomics cores, facilities, chemists, including our own lab. The question is, how do we help those people? We certainly do not help them having a plethora of different ontologies and saying, well, use that ontology, not that ontology, because they will not know, A, about these ontologies, and B, using just, say, the word blood plasma to a clinician is an oxymoron, because of course plasma is in blood. So they would never use that term. They would use plasma, just saying. So we need parsers. We do not need more schedules of how, you want us to put a term into a field, into a repository where we may not even end up landing there. We need parsers that work autonomously, right from whatever the clients give us. It's linked directly to understand the texts and understand the study design, and maybe ask another question, right? Okay, so you have told us about this and this and this, so I don't understand, just like artificial intelligence would do. And so that's what we need. And redo is the wrong approach. Not that it's anybody's fault. It's like you have to do when you have the data and you try to redo the experiment without having the actual biologist at hand. I have been to the Asiloma meeting last year, where actually the proteomics people from EMBL, they had a new person, and after 20 years, what he presented was just saying, well, he didn't understand the metadata and they didn't have a good ability to compare across the data that have been deposited for 20 years, mainly because of the missing metadata. So we have to, get the horse from the front, right? And, be able to grab the metadata right when we acquire the data, when we have the attention of the biologists.
Yeah, definitely. Yeah. The earlier the better, of course. Like if you are able to get the experimentalists via some tool to put right away the terms exactly how you would understand it, that's of course perfect. Large language models. I've tried some approaches. Like if you try this, for example, if you do some experiment, like for example, in metabolites, they have this like nice sections of like, here's our chromatography information, here's the MS information, here's the extraction information. If you pass that into, for example, ChatGPT and ask it to fill out some tables, it will sometimes work, sometimes it will work not so nicely. So, I mean, it's something that can be tried, and I'm sure that I know that you have done some work in that direction with language models and like filling out missing metadata fields. If it works, we are happy to implement it. Yeah, happy to talk later about that.
Can I also answer?
Oh yeah, sure.
I'm going to obey the rules and not just use my voice. So I agree completely, but both GMPS and us have to do what we get. But again, this is why we're here, to talk to you about what we do, to raise questions like this. Because I was at ASMS a couple of weeks ago, and really need to stop going across too many countries and time zones. So a lot of people came to talk to us about they were AI people or ML people, and their key message to us was that as repositories and as experimentalists, we need to help them to have the gold standard sets, and then they'll come and do the work for us. So again, this is an ecosystem. We all have to work together to make sure we can mine it effectively. And it was really nice what they had to say about the metadata in various resources. But again, it comes back to the experimentalists. And as my colleague said, we need to have the data in there to explore it. The other thing that we're doing in Europe under an Elixir infrastructure is pulling together vendors, because ideally neither you nor the curators should be putting things in Excel spreadsheets and sucking it into the database. What we would really love to have is coming out of the machines that we could parse it straight into the database in a structured way, and the pain points would disappear for a lot of us, and the learning would happen. Again, as Peter Dorenstein said, we have to interact with the companies, but if we build together as a community, we can make them do that. And there will be a meeting Q4 in Europe about that. And if you're from Europe, please come along to said meeting when we promote it and tell these vendors that this is what you want. Thank you.
Fred Tabong, the Ohio State University. Thank you for the great efforts that you're putting into this. I find this very useful. I was just wondering whether, as a community, we can develop a minimum set of metadata that would be required to even upload your data into one of these repositories. In a way that if one of those fields is missing, the data won't be able to be uploaded into the, but then there will also be, it would also be easy, to get help to fill up those required metadata fields. So I think that it is coming down to the metadata to make the data that is available useful, right? Because we need the metadata attached to the annotated chemicals. So having a minimum set of metadata fields, I think, to me is important. Thank you.
Yeah, I think most repositories already have that. Like they have some minimum set, like in metabolites, you won't be able to load anything up without adding certain metadata. In GMPS, you can. And I think I don't think we will put, I mean, I will also talk with the other people responsible there, but I don't think we will put like an absolute minimum that everybody always has to put. Because also if you don't have them, they can be quite useful actually. Like some of the things that we have published, we did that without the use of basically, even without the use of any metadata, you can already learn quite a bit. And so if some people are submitting data and they would not have, if they would have had to put the effort and you lose them that way, it's a trade-off. I'm not saying no, I'm just saying like there's things to consider for this kind of thing.
Yeah, so I think every repository has this sort of minimum metadata requirements, and so that's already in place, but it's different from each repository. Although GMPS, you can upload anything. We feel that it's the user choice how much they want to share or not. But if you end up choosing, let's say, the redo interface, you can actually upload any metadata into the redo table. There's no restriction. So in terms of vocabularies, you can then later parse it. But we do have a metadata checker, and that checks for minimum data, and that tells you that this metadata is compatible with the rest of the system and ready for data science. And so this is actually an automated checker, and it will tell you what parts of the metadata table needs to be corrected to be compatible. So that's how we did it. But there, in terms of metabolites, they have a manual curator. He's hiding here. This is one of the reasons why the metadata in metabolites is actually very good. I think Metabolomics Workbench also does a great job in terms of capturing the metadata, because you're not able to upload the data almost if the metadata is not there. And so you just have to, but each of them will have minimum metadata requirements. I do like what Oliver is saying, is that, you know, at some point there will be metadata that is just not structured, and there will always be you know, differences in how we write things. And we can write male, boy, girl, female, that sort of things. And parsers can really help with that, where we, you know, basically, it sounds like Metabolites has some examples of parsers that they're already pushing things through. But there basically needs to be more parsers. And some of the things that Yasin is doing, the harmonization of this metadata, it's essentially a parser, so that you can get the data ready for data science.
Yeah, I just wanted to reiterate what Claire said before. I think also, Oliver, in a way, it is a little bit unfair to put all the weight on getting the metadata on the repositories, where actually is the role of the field as a whole. So if we could get like LIMS systems that can output already the information, then it would be very easy for the repositories to get it. The same for analysis tools, that if you provide that information in advance, then it would be a trivial, I don't know, output or export or something like that, and then it would be easy to. I agree that of course a posteriori is much more difficult, but this is really the role of the field as a whole. It's not really in my view, it's not really the fault of the repositories who, you know, have limited resources, and it is key that the whole field realize, and the whole software ecosystem, the LIMS especially, and the analysis tools can output this metadata can be taken later by the repositories. For that, it would be nice to have like a common format to do that, something that we can talk about. But yes, just don't consider this is the kind of fault of the repositories. It's really the obligation of the field as a whole, that the LIMS systems, the analysis tools, at the moment that they take the data, this metadata can be there already, and then it can be exported. You just give a push in a button and say, oh, I want the metadata, and then, you know, this is the way. I think this is the only way to do it. And of course, AI can help a lot, hopefully, but I think that needs to be explored to really see how it works. And yeah, this is what I wanted to say.
So if I may share some experience along those directions that I have had, mainly playing the role of actually taking the metadata from all the studies in Metabolomics Workbench and depositing that metadata information into the common fund data ecosystem that I mentioned earlier. So as many of you who have looked at the study information at Metabolomics Workbench, you know that as such, behind the scene, we don't formally use ontologies like Oberon and so on, although we have tried to use a reasonably consistent set of terms. When I submit that metadata into the common fund data ecosystem, they allow only using ontological terms. They don't allow me to enter like something like blood plasma and so on. So I can leave the field blank, but if I enter there, I need to put an Oberon ID or some ontology term like that. So based upon what we are learning from that, we hope that down the line, we will be able to take that experience and put that into Metabolomics workbench in terms of at the metadata summation interface itself, allow this feature to let the metadata submitters choose the correct ontological term so that it will be consistent at that level itself. Now, of course, the challenge will be that in some cases, they are not able to select a correct ontological term that they might want to, and that's where we think that maybe more versatile systems like UMLS or Sunomid CT and related ontologies might be able to help, where even if you cannot find a precise term that you are looking for based on UMLS tree, you may be able to find something similar so that at least the ontological term that you use would be at least partially accurate. And I think those efforts, I think, I don't think it's going to happen this year or even probably early next year, but down the line we hope to make some progress on that front at Metabolomics Workbench.
Great. And thank you, Yashin. Let's proceed to the fourth talk by Kyobin-kan from Korea.
I do the file here, so if there's only one file in the history, so you can see. So, morning. I'm Kyobin from Sungkyung University, which is placed in Seoul, Korea. And actually, I think like most of you actually have never ever heard about our name in here. So actually, this time I'm gonna briefly introduce like what we started in Korea about the data repository of Bolt and what we're gonna do in the future or near future. So I'm gonna talk about this briefly. So, well, I'm going to introduce the Korea Bio Data Station at the first. So this is the comprehensive repository for all types of biological data, well, processed in Korea. So actually, Korean government established our own repo for any kind of data, including like genomics and proteomics, and the transcriptomics, and of course the metabolomics, and even like bioimaging data. So it's like a integrated system for any kind of data, and then basically it's designed for a multi-omics data. So it in its infrastructure, it has like the biosample and the bioproject structures, and then the same biosample can be like different types of data in the multiple types of data. So it's established just a few years ago, but now it's like still being in the process of optimization. But the Korean government, we're going to make it mandatory to put any kind of data coming from the project funded by the government. So we believe that we're going to be huge in the future. So so we have multiple different data set. Actually, repository is categorized based on the types of data. So we're a part of a Korea Metabolomics data repository. The abbreviation is KMAP. It's a little bit strange abbreviation, I think, but mainly it's because the proteomics repository pick up the name, the K-pop, as the Korea proteomics repository. So anyway, so it's a part of a KBDS. So both of animal and mass spec based data were going to be covered here. And we benchmarked a lot the metabolite when we first designed it. So mainly the metadata format or minimum requirement for like a metadata and the data deposit is really quite similar to the metabolite. And I think like the other the genomics or the transcriptomics data repo will gonna also be really similar with the ones in the EMBL. So anyway, I'm part of the Metabolomics Data Curation Center. It's quite a weird structure, but I think so the KBDS is mainly ran by Korea Institute of Biotechnology, and then biology and biotechnology, and mainly the guys in there are really expert in the genomics, but they do not know very much about the metabolomics and the proteomics. So this is kind of a center, isolated from the operator of the repo, but actually we took part of our role as the adversary panel for a repo. So it consists of expertise in the proteomics, and our center is in the have multiple metabolomics guys in Korea, so we just give a future direction and what types of modifications should be made in the repo, and also we're gonna take the role as the manual creation of metadata and maybe a lot of data in the future. So yeah, we do some types of jobs related to the metabolomics repo in Korea. So yeah, director is the Professor Ju, who's sitting in here, and then multiple PIs are in the center, and we tried to include the metabolomics persons in the multiple domains, so not just the human sample based on metabolomics, but I mainly I do the plants and the microbial, the secondary metabolite, and also we include some persons from the plant and the microbial metabolomics, included both of the primary and the secondary. So we're trying to make the repository can cover a global types of data. I'm gonna just pass the roadmap, and so what we have done. So the center was established in 2022, and we tried different types of work. So the first thing is that we made up the quality control guideline. Well, it's written in Korean, but mainly the goal of this one is it's a brief guideline to let the others. Others who want to start their own the metabolomics project here, or want to make up their own data and deposit their data in the KBDS, to let them know okay, you need to make up the pooled QC sample like this, or you have to just put the standard in this, and further for example, you have to take care about the metadata and what types of information should be included in the metadata. So we made up the guideline, and it's categorized for multiple different types of samples, the targeted and non-targeted. And in case of when we use the LCMS here or GCMS here, or even cases you need to when we do the secondary metabolite based metabolomics. So we write down, we are writing some, and some of the guidelines already published in our website. So yeah, we are trying to make up and enhance the quality of data produced in Korea by this effort. And the second one is, we are putting the reference data set into the KBDS. So mainly the human samples were being produced based on the prospective clinical trials ongoing, and then also we are producing high quality plant and microbial extract data set, and also we are putting them into our repository. And then what was it? So yeah, we're trying. This is actually a tricky part, but we are trying to make up some metric to estimate the quality of a data and the metadata deposited in the repository. So and also we are trying to training some people who are going to do the manual creation on the metadata, which will be deposited in this repo. And then in Korea we're doing a multiple workshop and some sessions in a conference in the first domain to promote like, okay, if we want to do the metabolomics, and then, maybe we need to care about this such kind of thing, like about quality control, using the QC sample, or maybe you need to ask the vendor to give you like a raw data file for open data science. So we're doing such kind of efforts. And then finally, we also trying to harmonize our data to the other repos. So because we are like a late starter, so it's quite convenient. Like we can just make our things to be harmonized to the other, the major 3 repos currently have massive data. So we're trying some efforts. So I'm gonna show you some the cases of details of this a little bit later. So actually, one of the reasons why the Korean government built our own repo is for this huge project. So there is a national project of bio big data in Korea. So the Korean government just investigated 0.45 billion US dollar for 5 years. So we're gonna build our own the biobank and the bio data bank. So we're going to sequence the DNA, and we're going to collect the medical cohort for 1 million population in Korea, including both of healthy and some patients having multiple diseases. And metabolomics we're going to also be included in this project, although it's not going to be really huge, but there is a scheme in the right side of the slide. Well, actually, unfortunately, the words are in Korean, but you can see that, we're gonna run like we're gonna gather tissues of the 6,000 cancer patients in our hospital. So, the malignant tissue and the normal, the healthy tissue are gonna be collected separately from the same individuals. I mean, like 1,200 per each cancer type for 5 different cancers. And then they're gonna be separated, like the homogenized, and they just separate into some tubes, and they're gonna be delivered to the labs, and then metabolomics data, and then proteomics data, and then the transcriptomics data are gonna be produced from the exactly same person, so same tissue samples. So, and then obviously DNA samples were going to be also sequenced by their own plasma, and then the plasma were going to be also kept in the biobank. So we expect that the multi-omics data were going to be a huge thing to be analyzed in the future, and it was going to be deposited in our repo within 5 years, we expect. And then also we want to make the metabolomics data repository to be harmonized to the others. So already Yashin highlights what actually we can do through the pan redo and the GMPS2. So we're building up the API services to make it possible to access to the Korean repository data through the universal spectrum identifier. I think it was gonna be done within this year, and then also we're gonna do the metadata harmonization for the pan redo, and also I believe that it will not gonna be difficult because our metadata format is really similar to the ones of metabolites and all the metadata. Obviously, of course we're gonna be in English, so we can just put our data there, and then we're gonna convert it to the compatible to for pan redo soon. And also, a third one, this is not about the data deposition, but the GMPS2 environment is actually trying to be delocalized in multiple different countries. And then already, so I contacted the Ming in the UC Riverside, and then we're building a delocalized server of a GMPS2 environment in the KVDS. Actually, they have a huge supercomputing resources then, and already they allowed us to use some parts of it for the environment. So just not keeping our data in our storage, we can also serve as kind of a environment for the data analysis and re-analysis of the data in the public repos in ours, and of course in the other repos through this. So thank you, and yeah, this is the end of my talk. So if you have any questions about it, yeah, please.
I have a question.
Yep.
And it goes towards the manual curation. And I've had discussions with the metabolites team on this before. But currently, I would say probably about 1% of the metabolomics data goes into the public domain.
Okay. Yep.
And if that becomes 100%, we need 100 of these guys.
True.
It's not going to be reasonable. I don't think any funding agency is going to support 100 people that are going to manually curate. So I found it. So why did you choose that particular strategy? Since you're creating a new repository, and not find other ways and ways where you can avoid and minimize the impact of personnel power, which will become the bottleneck. I don't know. I don't know. Maybe metabolites can say how long it actually takes to curate, but you can probably estimate your growth projection and how many people you'll need for manual curation.
So basically,
sorry. Actually, I love to hear from Tomas or Claire.
So we're not like a 100% confirmed about the curation strategy, but I think like a manual curation, maybe in the near future, it will not gonna mean that 100% manual. I think like it will maybe like a manual curation, but aided by the AI. Well, still, I think like manual check on for example, like AI aided the curation on the metadata still will be required, right? So I think like we can minimize the amount of work we need to do through like such kind of aided by like a technological aspect. I didn't mean like we're gonna just manually create the entire metadata. It was gonna be in here.
I'm so sorry.
Yeah, correct. It doesn't scale,
Yeah.
and we are facing these issues now. What's super important is that I get to talk to the software developer. So now we have now implemented a new granular study validation framework, which will be at point of access, and also you'll be able to run it locally. So we've made a big change, Peter. I thought Claire was going to announce it. So announce it. So yeah. Before, you had to have manual curation, and it does add a lot of value. With Yasin's work, like almost 100% of the metabolites data could be reused without manual intervention. But now, because of the scaling issues, there will be a new status for metabolites, which at the moment we have community consultation, but we'll call it public unreviewed. So as long as you meet the new study validation, which is granular, you can go have your public accession. You can get it within 1 minute, whatever time frame you want. But what is important for us is that there is this now consensus within the field that these are important fields at the sample level that we don't want to get rid of. So as long as you fill out these key fields, you can have your public accession. But yeah. Oh, Claire.
And the other key thing is, and this is what we will be exposing if you come to our poster, and what we want to talk about with our repository colleagues is those minimal automatic validations. And that's what, we're not going to dust it. That will evolve, and that's why we're here, and why we want to talk to you at our poster. And I didn't want to, this workshop is but we also want to talk about what Metabolites is doing, and then we'll bring that to our colleagues. again, we need you to tell us what's maximum impact for you. I'm so excited. I'm second guessing and chattering, but not quite.
Okay, we can go there.
From Japan about hand-creating repository. So thank you, Kevin. While preparing, I'd like to point out the problem of matter transfer example. In Japan, people talk about metabolomics in Japanese basically, and they don't read English pages before submission. For us, it's very important that our curators contact with some readers so that they can use proper name and proper style. And yeah, we are improving because instead of just submitting raw data to Metabolomics or MIC or Workbench, other repositories, we encourage our government to hire our own curators so that we can. I think this trend is still high. Yeah, I know this trend should be broadened to other continents like Latin America or Africa. So as you think about human personal genomes or personal metabolome, obviously that each country wants to establish own repository with own curators. And what's important is to harmonize those established repositories to use the same standards, same nomenclature. So it's ready. So let's continue.
Morning, everyone. So I talked about Metabobank. It's a public metabolomics data repository in Japan. So first I talk about the overview of the Metabobank datasets and function and some history. And the main topic is, so of course I want to promote data sharing of the metabolomics because I am a metabolomics researcher about 20 years ago, and I'm making many databases or tools for the metabolomics community in Japan or also in the world, so that I talk about the three topics today. And I want to know how to promote metabolomics data sharing. Okay. So this is a top page of the Metabobank. So in Japan, some metabolomics databases existed. Of course, now exists in some dataset created by some organization or laboratories. And in Japan, the Japanese, so we want to integrate to that dataset to the public repository. So we made the Metabobank at 2020. So it contains mass spectrometry or an NMR data or imaging data. So the submitter can use this page for the data submission. It may be so. Oh, sorry. Look like this. So from this data submission tab, and the users who want to download or to check the dataset, so from here the search and download data is available. So, and in case of the metabolomics data of the human individuals in Japan, so there is so the datasets must be deposited. The raw data is must be deposited in the control access database, like JGA is hosted also in DDBJ. So, and in this year, so we realize publication of the NAR database issue. So the details is described here. So this is a brief diagram of our database site. So there's a search function. So from here, and the list, you can see all data sets. There's an ID of our database as the ID. And you can check, you can filter out the data sets using the instrument, GCMass or LCMass, etc. So, sorry for it is not visible in this slide. And you can filter out the organism from here, Homo sapiens or Arabis thaliana, etc. here. And this is, if you click the study ID, so you can see the summary of the study. It looks like metabolites. So some description and the title, and you can download from the all data from here. And also there is some bulk download page from here. So this is a list of the study IDs, and if you click, you can download all data from here. And now the Metabank contains 138 studies, about 1 terabyte files. And this last year, so we include the metabolites metadata from here. So we index the metadata of the metabolites. It's now 1,500 studies here. So, and we want to add index of the metadata of the workbench or GNPs, or of course, there are more we want to do it. So this is statistics of our data sets now. So, and the data sets increasing. This is the number of the study, and this is the number of the organisms deposited in the Metabank. So it is increasing every year. And this is a breakdown of the data sets. So it is an instrument. So the half is LCMass, and about half is also GCMass. And NMR or mass imaging is a few data sets. And this is the organism. So in Metabanks, 80% is from plants, and 10% is animal, and others, microbe or human is here. So maybe. So please access our website from here, the Metabank. So next I talk about this year's, last year's or this year's new feature in the Metabank. So I have questions. So how many people know the bioproject or biosample? I think okay, thank you. So it's very few. So bioproject and biosample is one of the database. I think started from the USA, NCBI. So it's the main purpose is to sum up the genome sequence or transcriptome data sets. So the bioproject contains omics experimental investigation information is included in the bioproject, and the biosample contains sample information to use the omics experiments, genomics or transcriptomics. So the organism name and the taxonomy ID, or when the sample is collected, or where the sample is collected, or etc. And also the bioproject information contains submitters information, organization or who do it, etc. So, and in Metabanks, registration of the bioproject and the biosample is mandatory now. And so the DDBJ, so my organization is hosted the sequence or transcriptome database is as a public repository in Japan. And the transcriptome database in DDBJ also used, and also genome database used the bioproject and the biosamples. So, so it's easy to summarize the validator or some rules in containing the Metabolomics database. So we use it. And so the merit of the bioproject and biosample is it is easy to manage related omics data sets, the genome, because there are many publications using transcriptome and metabolomics in both. so to do the function dynamics or to explore the new pathways or new molecular markers, etc. So I think it is convenient to catch up and manage this. And so now the Metabolomics data can search from the by project database from like here. So I think I want to metabolites want to use a biosample. I don't know, but I heard. Okay, so the next topic is IDF and SDRF metadata formats. So of course, metadata format is depend on each repository. So, but in most cases, it's something like tab text format or text format. So we compare many data formats when we created the database. So we have done some trial to brush up each formats, and finally we decide to use these formats. So it's like the tab text format, like Excel file. So I think it is easy to fill in the formats for submitters. So and this format is compatible for the ISA tab in Metabolites. So and the IDF contains investigation information, and SDRF is sample information, sample and data relation information. So IDF contains study overview, experimental design, protocol, publication, and submitters information, author name or organization name. And SDRF contains sample characteristics like tissue name or organism name, etc., and also relationship between sample and measurement and instrument type and data file name, etc. So like this. And we make the validator and control vocabulary 100 terms, and it is freely used from our Metabolomics sites. So IDF and SDF is based on the MAGE tab format. I think it's first used or used gene expression database in EBI, I think, and and now we use MAGE tab format in gene expression database in DDBJ Japan, and also I think I heard the proteomics database JPOST in Japan is used also these formats now. And third topic is raw data format, MZP. I think someone know these formats? Oh, I think, it's okay. So this is a new format, so it's not unfamiliar for all, I think most of people's. So the data format is basically in Metabolomics, basically it's highly diverse. Of course, there's many formats for the vendor specific in each vendors. And also there is some open standard formats like MZXML and MZML and so on. So in Metabolomics, the submitted raw data file is no restriction. So any format is acceptable because the regulation is very difficult now. I think it's same as the repositories. But this is, of course, this is inconvenient to reuse, to reanalyze Metabolomics data in bulk. And also, it is inconvenient for the curator itself, himself. So we want to check the raw data for the submission time, and we need to convert some format, the vendor specifics to text XML or etc. So to browse the data is correct or well something. So, and we have collaborated company, LifeX. I think so, LifeX from here. Yeah, here, let's see. So we start collaboration to support MZP low data format. It is a complex, less binary format, I think, and it's a very small size. And the browser software is freely available from the company site. And so it enables to reduce the curation cost for our curators. And also we would share the NSB raw data format files from our sites. And it's just this June. So, and the poster is 1021, so please visit if you want to need to know the details of the format or etc. And this is the data checker is a free software to browsing the MZP format file, and we convert some raw data or something GCMS data into the MZP format and provide from our Metabank sites. So it's just a future perspective. So, so in Metabank, only one curator exists. So it's so the submission is increasing every year. So we must make a better registration process to make more better validation programs, or some, of course. So of course we must decide which metadata is more important for the community, because the curation cost is very limited. So we must focus some terms if the term is more required for in the communities. And of course, we must brush up our search function. So especially for the chemical names or the spectral, etc. But I think it's something feature, because of course it's difficult compound name problems, synonyms. It's a very tough job for to developers, and of course in the mass of course there is many programs freely available, or some system is existed, but to implement the database is very high cost. So, but we want to try it, and of course, and finally, so we want to link the metabolics data to the other omics data like genomics and transcriptomics, etc., and also the bioresource information, or maybe biodiversity information, because it's very important in the world. So to prevent species, to protect the species, or the live resources for the human society, or for the health, or something like this. So it's collaboration, so many people under the grant. So thank you for your attention.
Hello, thank you for your talk and for this introduction. You mentioned that your data is isotype compatible, or that so there's a link there. Does that mean that if someone makes an export that is, let's say, ready to be uploaded to Metabolites, for example, you can take it directly, or you still need to tweak a bit? You still need a special adaptation for Metabobank?
A little bit, I think. Mm-hmm. I think that. I think decrease it, not too much, I think.
Mm-hmm. Okay, thank you.
Okay, any more? Oh, okay.
So you talk of multi-omics. Actually, metadata in multi-omics is super important, more important than just in metabolomics. I wonder how you guys are dealing with that. You said biosamples, but could you elaborate a little bit more about metadata requirements for linking different data types?
So of course, of course, it's very important to multiomics studies, so to integrate the metabolomics and others. So, so bioproject and biosample is, I think, one of the solutions of this. But of course, this metadata is some of regulations, so it might be so. But it is used in USA and EU or some Korea and Japan. So but there is some minor modification in each repositories. So, but in most cases, I think the trend of the making the bioproject biosample system is gradually increased. I think this decade, 10 years or more. So, and because there are many omics studies, proteomics, phenomics, or others, toxicomics. But I think it is better to integrate all omics information to the something like some simple system. So we recommend this solution, but of course we need to try to use it in the community here.
Okay, more questions, let's discuss after the session. And thank you for gathering to this workshop.
Thank you.